We have been discussing meiosis now for the last several lessons, and now it's time to get down to the nitty-gritty of exactly what happens in each phase of meiosis. So this is part 6 of Unit 4, and we're covering pages 205 to 208. So let's get started. First of all, an overview. Meiosis reduces the chromosome number from diploid to haploid. We discussed what that means previously. Meiosis involves 2 consecutive cell divisions. So again, that's 1 cell dividing to make 2, and then a second round where the 2 cells divide again to make 4. So we end up with 4 haploid, genetically unique daughter cells, which can then be used for reproductive purposes, either as spores or gametes. Remember, plants and fungus use meiosis to make spores. Animals, of course, use meiosis to make gametes, sperm cells and egg cells. So this is just kind of a quick sort of pictorial overview of what's going to happen. Prior to meiosis, every chromosome will replicate during the S phase. Remember, G1, S, G2. So during S, the chromosomes will replicate so that each chromosome now will have 2 genetically identical sister chromatids. They start out genetically identical. They do not end up that way, as you will see. But in the first half of meiosis, the pairs of chromosomes separate and end up in separate cells. So remember, each of these cells is a haploid cell. Each of these cells will have half as many chromosomes as the original cell. And then there's another round of meiosis, the second half of meiosis, where each of these cells splits again, and we end up with 4 haploid cells, each of which has half as many chromosomes as the original cell, and 1/4. If you compare this cell to this cell, each of these 4 daughter cells has half as many chromosomes and 1/4 as many chromatids, right? Because if I look at this picture, 1, 2, 3, 4 chromatids in this cell, but when we're finished, notice there's 1/4 as many chromatids. Wow, so it's like mitosis, but clearly it's a little more complicated, and we're going to figure it all out. So let's begin with prophase 1. You have to put a 1 beside it because remember, everything happens twice in meiosis. So prophase 1 of meiosis, this would be after G1, S, G2, and now it's ready. We're ready for meiosis to begin. So this would be either a primary spermatocyte in male animals or a primary oocyte in female animals. And notice what I've done here. I'm making it so that we can compare prophase of mitosis with prophase 1 of meiosis. So I don't think there's any better way to fully appreciate the difference without just looking at it. So if we're talking about humans, remember there would really be 46 chromosomes in this cell, and that would be 23 blue chromosomes and 23 red chromosomes. But of course, they're only showing 4 chromosomes, 1, 2, 3, 4 in this picture. And if I go down to my mitotic prophase, again, 4 chromosomes. They're not color-coded, but it's the same 4 chromosomes. Chromosomes come in different sizes, so you notice we have a pair of large chromosomes and a pair of smaller chromosomes. And if you recall the picture I showed a couple lessons ago of the human karyotype, where we could see all the chromosomes sorted by size, they come in different sizes, small, medium, and large. But so the point is there would be 46 chromosomes if this was a somatic cell. There's 46 chromosomes in the primary spermatocyte or primary oocyte, as the case may be. But right off the bat, you notice a big difference in how the chromosomes behave. In mitotic prophase, when the chromatin coils up to form chromosomes that we can see and count, the pairs of chromosomes don't really act like pairs. The two big chromosomes don't pair up, they don't come together. The two smaller chromosomes that clearly go together, they don't act like a pair, they just ignore each other. It's like 46 chromosomes that really don't associate with each other. That's very different in prophase 1 of meiosis. Every chromosome pairs up with its homologous partner. So if this is a small chromosome, it will pair up with another small chromosome. This was the one that was inherited from the dad. This is the one that was maternal, inherited from the mom. And they'll pair up with each other. And they come together physically, and they act like a pair. They act like a couple at the prom. That's the way I always think of it. And just like couples at the prom, sometimes they get a little handsy. They can't keep their hands off of one another. You notice that the red chromatid and the blue chromatid are touching. They're physically connected. And there's a really important reason why that happens. That coming together of pairs of chromosomes is called synapsis. And it simply does not occur in mitotic prophase. So let's look at it here. Again, prophase 1 of meiosis. This is after interphase. So we already had G1, S, G2. Now meiosis has begun. And we have synapsis, the coming together of chromosome pairs. Of course, we're going to have typical prophase events. I mean, the chromatin coils up to form chromosomes. The nuclear envelope breaks down. The centrosomes begin moving apart, the spindle fibers form. All of those normal prophase events happen. But then you have this very unusual situation where the homologous chromosomes pair up. Each pair of chromosomes is called a tetrad, which is kind of an odd term because tetra means 4. This is 2 chromosomes, but it's 1, 2, 3, 4 chromatids. And the other thing that's really important that happens here, I mentioned how touchy-feely they are. The non-sister chromatids, the red and the blue chromatids, overlap and touch each other. And every place where they touch is called a chiasma, chiasmata plural. And the reason they do that is because a piece of that red chromatid and a piece of that blue chromatid will break off and they switch places. This is crossing over. This is the exchange of DNA between a paternal and a maternal chromosome. So it's pretty cool, pretty darn important. Let's summarize the events of prophase 1. So we have typical prophase events, chromosomes coiling up, spindle fibers forming, nuclear envelope disappears, nucleolus disappears, all that normal prophase stuff. Then we have synapsis, the coming together of homologous pairs of chromosomes. And every time a pair gets together, a blue and a red chromosome, we call that a tetrad. What is a tetrad? It's a cluster of 4 chromatids when a pair of chromosomes, a blue chromosome and a red chromosome, come together. Every place they physically touch each other is called a chiasma, chiasmata plural. And while they are touching, they swap DNA with each other. The red and blue chromatids exchange DNA. This is crossing over. So it's, I want to make sure we understand the significance of that. Remember, if you're making sperm or eggs, this is your cell. These are all your chromosomes. But where'd you get them from? Well, you got half of them from your dad, and you got half of them from your mom. Before you pass these chromosomes down to your kids, before you pass the chromosomes down to your sperm cells and your or your egg cells, whichever you happen to make in your body, before you pass them down, what are you going to do? You're going to shuffle the paternal, that's DNA from dad, and maternal, DNA from mom. You're going to shuffle them together. So later on, when these pairs separate, you notice the red chromosome is going to have some blue on it, and the blue is going to have some red. Every single chromosome will eventually have a mixture of paternal and maternal DNA. You're literally shuffling the DNA that you inherited from your two parents. You're shuffling those genes before you deal them out to your offspring. All right. So this is another illustration of exactly what crossing over accomplishes. Here is a tetrad, a pair of homologous chromosomes. During prophase 1, they begin to swap DNA back and forth. And after metaphase 1, when they separate and eventually they end up in separate cells, notice the blue chromosome has some red on it and the red has some blue. And this is a random shuffling. It's different every time. So every time sperm cells are made, every time egg cells are made, the shuffling is a little different. And so you always end up with a unique combination of maternal and paternal genes in the gametes. And then this was my attempt to illustrate this, so it's not quite as colorful, but I just used white and black to show the maternal and paternal chromosomes. And so if you put genes on here, and of course each chromosome can have 1,000 or more genes. I'm only showing 2 genes. And again, I'm using the same example I used before where capital A represents an allele for tall and lowercase a is for short. So maybe my mom gave me a gene for tall, my dad gave me the gene for short, mom gave me a brown-eyed allele, dad passed down blue eyes to me. Well, now these are both my chromosomes, right? I inherited them from my two parents. But before they get passed down to gametes, before I pass these genes down to my offspring, what am I going to do with that maternal and paternal DNA? I'm going to shuffle it. That's what crossing over does. So notice, if you look up here, before crossing over, the only 2 things I could pass down to my kids would be a tall gene attached to a brown-eyed gene, or I could pass down a short gene attached to a blue-eyed gene. Two possibilities. But after crossing over, if you look at the same 4 chromatids, see, this is 1, 2, 3, 4 chromatids. After crossing over, at the end of meiosis, each one of these chromatids is going to end up in a separate gamete. I'm using sperm cells as my example. So this chromatid is going to go to this sperm cell, this chromatid is going to go to this sperm cell, this chromatid goes to this sperm, and this chromatid goes to this sperm here. But notice they each have a different combination. This is tall with brown eyes, so I could pass that down to my kids. This is tall with blue eyes. I could pass that to my kids. Short with brown eyes, short with blue eyes. So there's 4 different combinations that I could pass down to my kids. Before crossing over, I only had 2 combinations, either big A, big B, or little a, little b. So that's the importance of crossing over. And it's important to note that I'm only showing 1 pair of chromosomes in my example. How many pairs of chromosomes do you have? 23, right? 46 chromosomes, but that's 23 pairs of chromosomes, 23 white chromosomes and 23 black chromosomes, right? So that's 46 chromosomes, 23 pairs. I'm only showing one pair. So just imagine all 23 pairs are doing this. Think of all the mixing and matching that's going on. And I'm only showing one crossover event between these two chromosomes. I'm only showing one place where the DNA breaks and swaps places. The reality is you would have multiple crossover events, maybe 20, maybe 100 swaps back and forth between these two genes. And I'm only these two chromosomes. I'm only showing two genes. The reality is there would be over 1,000 genes on this chromosome. So the number of possible genetic combinations created by crossing over it's a huge amount, a huge number of possible genetic possibilities. So let me back up. So here we were, prophase 1, that's when crossing over takes place. And then, let me get to the right place. Then we go to metaphase 1. After prophase 1, it's metaphase 1. And you know what happens in metaphase. Always, right? Spindle fibers attached to the chromosomes, pulling in opposite directions. And where do the chromosomes end up? They line up in a plane across the middle of the cell. Now, in mitotic metaphase, how did the chromosomes line up? Single file. This big chromosome is not paired up with its partner. The little chromosome is not paired up with its partner. It's like a conga line at a dance. Nobody's really acting like couples. They're just a single file line. But in metaphase 1 of meiosis, it's couples at the prom. They're on the dance floor. They are inseparable. And even if you get them in a straight line, they're lined up as pairs. It's not a single file line. It's a double file line. Because you know how couples at the prom are. They're inseparable. They're touchy-feely. They started this hands-on approach in prophase 1 where they just couldn't stop touching each other. They've already done the physical exchange of DNA, so the crossing over has already taken place in prophase 1, but they're still physically kind of attached to each other. So when they line up in metaphase 1, they line up double file. So the chromosomes line up along the metaphase plate. That's just a fancy term for the equator of the cell. But how do they line up? In pairs. Double file instead of single file the way they did in mitotic metaphase. After metaphase 1, it's time for anaphase 1. Now you remember from mitosis that anaphase, ana means back or opposite. Anaphase is where in mitosis, anaphase is where the chromatids separate. So each chromosome splits in half, and one copy of each chromosome goes to one end of the cell, the other identical copy goes to the other end. So in mitotic anaphase, the sister chromatids separate. That is not what happens in anaphase 1 of meiosis. Remember how they were lined up. The chromosomes were lined up still as pairs. So what's going to happen is one chromosome from each pair is going to be pulled to each end of the cell. In other words, the couples separate. So I always say anaphase 1 is like, and this happens a lot at the prom. I've chaperoned a lot of proms in 24 years of teaching, and I can tell you there's always a point just about every couple has a fight. I don't know what it is about the emotions of prom, but they're touchy-feely. They can't get enough of each other. They're just all over each other on the dance floor, and then at some point, usually the guy says the wrong thing, or looks at another girl or whatever, and they have a big fight, and so they go their separate ways. And they might have ridden together to the prom, but they might not necessarily leave the prom together. Anaphase 1 is the big fight. Okay? In fact, that makes a great extra credit phrase that pays. Anaphase 1 is the prom night breakup. Anaphase 1 is the prom night breakup. Each pair splits and is pulled apart. Let's summarize that. In anaphase 1, spindle fibers pull the chromosomes towards opposite poles. Homologous pairs are separated. But notice what does not happen. The sister chromatids do not separate. Let me go back to our comparison. In anaphase of mitosis, the sister chromatids separate. But in anaphase 1, that doesn't happen. Notice this chromosome still has both of its chromatids. This chromosome still has both of its chromatids. Both of its chromatids. Both of its chromatids. The sister chromatids remain attached in anaphase 1. Now, just about anything that happens in a cell can mess up on occasion. Mistakes happen. And it is possible in some cases where you have a couple who doesn't break up. Sometimes, instead of the two chromosomes going their separate ways, sometimes both chromosomes, let's look at this picture, we can see it a little better. So what's supposed to happen is the couples line up double file, metaphase 1, and then they have the big fight, so each couple splits up and they go their separate ways. But notice this couple didn't split up. Sometimes a homologous pair stays together and both chromosomes get pulled to the same side of the cell. See, both of these are going to the left, and what's supposed to happen is one of these should have been pulled to the right. So if you have a couple that doesn't break up, that's called nondisjunction. Failure to separate. May not seem like a big deal, but remember, this cell is going to split into two cells. So because of nondisjunction, this cell is going to have one too many chromosomes, and this cell is going to be missing a chromosome. So you're going to end up with sperm or eggs that either have too many chromosomes or they're missing a chromosome. That's called nondisjunction. It is the failure of homologous chromosomes, usually just a single pair of homologous chromosomes, failure to separate. There's a couple different places this could happen. It can also happen during anaphase 2, but we're looking at it here in anaphase 1, where the homologous pairs are supposed to separate and go their separate ways, but this pair failed to separate. So what's the result of that? Sperm cells or egg cells that either have one too many chromosomes or missing a chromosome. And one of the most famous examples of this is Down syndrome, sometimes known as trisomy 21. Here is a human karyotype of a person who suffers from Down syndrome. Notice two chromosome number 1s, two number 2s, two number 3s, two number 4, two of each chromosome, except chromosome number 21, there should be 2 copies, but there's 3 copies. Trisomy 21. Now, how does that happen? Usually this is the result of nondisjunction during egg making. So you have a failure of chromosome 21, the pair of chromosomes fails to separate. So 2 copies of chromosome 21 end up in the same egg. So that's 2 copies of chromosome 21 in the egg. And then here comes the sperm. Of course, it has another copy of chromosome 21. And so the baby inherits, typically it's 2 chromosome 21s from the mom and then a 3rd copy from the dad. And so that's incorrect. That's a total of 47 chromosomes instead of the normal 46. And that creates some genetic imbalances that affect the development of certain parts of the body and creates the problems that are sometimes associated with Down syndrome. There are certainly other chromosomal disorders, but Down syndrome is the one that's kind of most common, and people tend to know more about it than others. After anaphase 1, the next phase is telophase 1. You can see cytokinesis beginning here, and it's a little hard to tell from this comparison, but the biggest difference is in telophase of mitosis, one copy of every chromosome is in this nucleus, and one copy of every chromosome is in this nucleus. So that's 46 chromosomes in this nucleus, 46 chromosomes in this nucleus. But in telophase 1 of meiosis, it would be 23 and 23. Remember, it's reduction division, so the number of chromosomes is getting ready to be cut in half. When this cell fully splits into two separate cells, each cell is going to have half as many chromosomes as the original cell. So this is telophase 1. This is a really important step in meiosis because this is where we go from a diploid cell. When it divides, we're going to now have two separate haploid cells. Telophase 1, quick summary, one member of each homologous pair reaches each pole, each end of the cell. A new haploid nucleus forms at each pole, so that's 23 chromosomes, and at each end of the cell. And when that cell divides by cytokinesis, we're going to have two haploid daughter cells. Here they are, two haploid daughter cells. They each have half as many chromosomes as the original cell. Whoops, wrong way. Now, in some species, at this point, after telophase 1, after the cell splits, there might be a pause, and the chromosomes might uncoil, kind of go back into a chromatin state, and it might be a while before meiosis 2 begins. Remember, we're only halfway through meiosis at this point. So in women, for example, in egg making, usually there's a pause after telophase 1. In men, in spermatogenesis, it's pretty much a continuous process, so you go directly from telophase 1 right into prophase 2. So there may or may not be a pause, but one thing is for absolute certain, there's going to be no further copying of the DNA. That only took place way back in S prior to prophase 1. That's the only time the DNA is ever copied, is during S. Okay, so at this point, you're probably looking at the clock and you're thinking, good Lord, Cavness is a half hour into this lesson, and we're only halfway through meiosis. When is he going to shut up about meiosis? And it looks like we've still got a long way to go, but really meiosis 2, the second half of meiosis, is much simpler. From this point on, meiosis 2 is really very much just like regular mitosis. Except that now our cells are haploid, but everything that happens to the cells from this point on is pretty much just like regular mitosis. So after telophase 1 and after cytokinesis is complete, we have two cells. These would be, in animals, these would either be secondary spermatocytes or secondary oocytes, right? And so there's two cells. And so everything that happens in prophase 2, metaphase 2, anaphase 2, it happens twice. It's happening simultaneously in this cell and this cell at the same time. So both of these cells now are in prophase 2. What happens in prophase 2? Well, typical prophase events. Right, so let's go back to the picture. The nuclear envelope breaks down. The chromosomes coil back up. If if they ever uncoiled. Sometimes they don't even uncoil after telophase 1. The spindle forms again. The centrosomes move to, eventually to opposite ends. So it's normal prophase events. But notice what it says here. In prophase 2, and this is important, there's no pairing of homologous chromosomes, no synapsis, no chiasmata. Remember, that's when the blue and the red touch each other. No crossing over. Why not? If you look at the picture, it's obvious why not. Because remember, when chromosomes get together as pairs and they swap DNA with one another, they can only do that with their homologous partner. In other words, if this big chromosome is going to exchange DNA with somebody, the only chromosome it can pair up with is this chromosome. And they used to be together in the same cell back in prophase 1. In fact, they were in the same cell up until just a moment ago when that cell split. But see, there's no way this chromosome could physically pair up with this one because they're in separate cells. And if this little guy wants to do crossing over, his partner is right here in the other cell. So the reason you don't have crossing over and synapsis in prophase 2 is because the homologous pairs of chromosomes are no longer together in the same cell. They are in separate cells. So prophase 2 is a lot simpler than prophase 1. After prophase 2, the chromosomes line up along the middle of the cell. You know, metaphase, they always line up. That's easy to remember. In metaphase, the chromosomes always go to the middle, and they're being pulled in opposite directions by the spindle fibers. And at first, they just form a line right across the middle. But notice metaphase 2 looks a lot like mitotic metaphase. Remember back in metaphase 1, how did they line up? Double file, right? They were couples at the prom before the breakup. And they couldn't get enough of each other. They were together as couples. Not in metaphase 2. Remember, the couples have already had the big prom night breakup, and so they're in separate areas. They're not even together on the dance floor. They're in separate cells. So they line up single file in metaphase 2. Metaphase 1, they line up double file. Metaphase 2, it's single file. After metaphase 2 is anaphase 2. Now, if you go back to anaphase 1, remember the sister chromatids did not separate. The homologous pairs separated, but the sister chromatids remained connected. So anaphase 2 is important because this is the first time in meiosis that sister chromatids, this guy and this guy, separate and get pulled apart. And an important point that I want you to notice here, if this was mitosis, if this was mitotic anaphase, what do we always say about sister chromatids? They're genetically identical, right? They're exact copies of each other. Well, that's no longer the case. In anaphase 2, when the sister chromatids separate, you can tell from this picture they are not genetically identical. This red chromatid has a little bit of blue on it, and this blue one has a little bit of red on it. Why is that? Well, that goes all the way back to prophase 1. Remember crossing over, where the blue and the red chromosomes paired up and swapped DNA with one another. So in anaphase 2, when the sister chromatids finally separate, they are not genetically identical. Anaphase 2, quick summary, the centromeres split. Remember, the centromere is that little circular button, and it's actually a piece of DNA that holds the two chromatids together. It splits right down the middle, and that allows the chromatids to separate. Sister chromatids separate in anaphase 2. And as soon as they separate, we don't call them sister chromatids anymore. When they're connected, they're called sister chromatids. But as soon as they're pulled apart and they start moving in opposite directions, we call them daughter chromosomes, and they are non-identical daughter chromosomes. Because of crossing over, these daughter chromosomes have different combinations of genes. You can tell from this picture they are no longer identical. And those non-identical daughter chromosomes are pulled apart in anaphase 2. Next comes telophase 2 and cytokinesis. Remember, cytokinesis is when we start to pinch in. These are animal cells, so they pinch into, and so this cell is going to split into 2 cells. This cell is going to split into 2 cells. You already know we're going to end up with 4 because meiosis produces 4 cells. And if you look at the color schemes, you can tell that each of these cells has a different genetic makeup. If it's like back in prophase 1, you took a deck of cards that you inherited from your dad and a deck of cards that you inherited from your mom. And in prophase 1, you shuffle those 2 decks together, thoroughly shuffling and mixing the genes that you got from your 2 parents. And now that you have dealt those cards out to your potential offspring, your sperm cells and your egg cells, no 2 hands is going to be the same. You know, if you deal 4 different hands from a shuffled deck of cards, you're not going to get the same combination of cards in any of those 4 hands. So that's kind of just a metaphor for this. I don't know if that's helpful or not. But you end up, let's do a summary of telophase 2. We're going to have a new nucleus on this side, and same thing down here. Brand new nucleus here, brand new nucleus here. Okay. Cytokinesis is going to split the 2 cells to make 4 cells. So we're going to end up with 1, 2, 3, 4 cells. And what is the final result? 4 genetically unique haploid daughter cells. In animals, these are gametes. These are either going to be 4 sperm, or if it's oogenesis, it'll be 1 egg and 3 polar bodies. Or, of course, if this was plants, these would be spores. 4 genetically unique haploid daughter cells that can now be used for reproduction. And that, I believe, is a good place for us to finish, because we are finished with meiosis. And don't forget to email me with the extra credit phrase that pays. I hope you have a great day.